Hello friends, we are going to discuss about different airborne bacterial diseases today. In our first topic today, we will be having a general idea and we will also like to know about whooping cough, a typical bacterial airborne disease. Now, man's life support system can be described as the Panchabhutas, where we can see that we have the Akash, Prithvi, Jal, Agni and Vayu. Vayu or air basically is very important, and at times the air has been mentioned as the breath of life. Actually, if there is a pleasant air, cool breeze, it relaxes us and makes us comfortable. While on the other hand, if the air is foul smelling or has some kind of pathogenic microorganisms in it, it brings us distress and can be illness causing or disease causing. Actually, we see that air behaves as a very good media for transmission. We inhale approximately 10,000 to 20,000 liters of air per day, which may contain about 10,000 and 10 lakh microorganisms. Some of them can be very pathogenic. Now, actually, what happens when a normal person breathes, that is, it inhales or exhales, and an infectious person breathes? Now, often when an infected person breathes, the air that comes out carries a lot of matter along with the air, and that may just be enough to cause infection for a susceptible person. But for an airborne disease, we normally see patients often coughing or sneezing, and these are the major causes for generating the droplet nuclei. What are actually droplet nuclei? Now, when the air is exhaled, at times water is also coming out with it and mucus may also be present. And we see that these, when they come out into the air first, may rest containing pathogenic microorganisms, which may be viruses, bacteria or even fungi. These are usually 10 to 100 micrometer in size, while after drying they generally assume a 1 to 4 micrometer diameter, which we generally know by the name of aerosols or droplet nuclei. Now, a person can actually even transfer or transmit airborne pathogens not only by coughing and sneezing, but also by laughing or common talk, or it can be even common contact. Very close personal contact can also lead to infections from the airborne disease transmission. Now, let us actually understand the respiratory tract briefly to know how this air, which is laden with pathogens, can enter and actually cause disease. Now, basically, we see here that the respiratory tract can be divided into an upper respiratory tract consisting of nasal cavity, pharynx and larynx, while the lower part, the lower respiratory tract consisting of trachea, primary bronchi and the lung. Now, actually, the upper part is in close contact with the air outside through the nostrils and can allow the entry of host of pathogens into it. While basically all throughout we will see that the surface area, especially at the lower level, are too much because these are actually meant for gas exchange. Isn't it? When we respire, we take in oxygen which needs to be given to the blood, and blood gives its deoxygenated air which is taken by this system and given out in the form of CO2 to the outside environment. So a large surface area is very important. Now, this on the other hand actually also helps in the establishment of infection by organisms which can be in numerous numbers over a wider surface area. Now, on the third part, the respiratory and circulatory system both are well connected due to this blood detoxification part, and that is why this can also introduce the organisms which are carried in through air to be transmitted to the bloodstream, so that the infection may not be restricted only to the respiratory tract but may also be systemic in nature, which means it can spread throughout. Now, we have seen how exactly the organisms or pathogens which are present in air can be transmitted to the upper and lower respiratory tract by the airborne transmission. But our system, our body will also provide some kind of a defense mechanism. Now, what happens? There is a ciliated lining in the bronchus region, typically in this area, beginning of the upper respiratory tract, and they start slowly reducing in numbers, and you have the presence of alveoli here where there are actually no ciliated cells, ciliated epithelial cells. Now, these cilia, what is their function actually? They beat, continuously beat, and do not allow particles to come down. If anything particulate matter, be it microbe, comes down along with air, they are pushed up. And at times, you know that we cough and throw out things at times. So this is what actually happens. But in the lower part, we have the presence of alveolar macrophages. And these are the immune defense cells of the lower respiratory tract. So although cilia will do the work in the upper region, in the lower part, if at all some organisms come, they will be killed by the alveolar macrophages. So our system, the human body, also offers some amount of defense against these organisms which can be pathogenic and borne by air.
Ma'am, does pollution aid in the transmission of airborne diseases?
Yes, pollution actually is not in direct relationship with our airborne disease, but it can trigger. It can also enhance the course of disease. It can change the course of disease because they may be finely related to asthma and allergy causing agents. The pollutants may be allergenic in nature, and they in fact at times are also called as aeroallergens that may transmit some form of allergy which may again be involved in the respiratory tract infections. Okay, so much so we have seen that both ways the pathogens are trying to enter the body while the host is giving some defense. So who will win actually? The pathogenic organisms, because we see in and around there are so many different types of airborne diseases, and we have some to list. But before we move on to that, let us see how we can go for some respiratory etiquettes, in the sense how we can avoid the transmission of such diseases. So there are a few tips which we can always follow. During cough and sneeze, we can cover it with the elbow or a napkin or a handkerchief, which can be disposed easily into bins and not thrown here and there. You can be at times asked to wear a mask if you are suffering from some respiratory ailments, and you may also be asked to sit in a corner called as a cough corner, basically trying to avoid contact with normal person or a healthy person. Now, after knowing the respiratory etiquette, let us see some of the diseases, the bacterial diseases, which we are laying more stress here. Here we have a list of diseases which are directly spread from person to person by airborne transmission. And the first in the list is whooping cough, which we will be discussing in detail today, which is caused by the organism Bordetella pertussis. We have several others in the list: meningitis, which can be of several types, pneumonia again can be of several types, and tuberculosis, not to mention, is very important, is also an airborne disease. Now, second in the list we have the second type. Here the organisms are directly not inhaled as such. The organisms do not enter through inhalation proper, but may come into contact of the respiratory system through other means. In the sense, let us take the first example. Psittacosis is a disease which is caused by Chlamydia psittaci. And how is it caused? From the dried or powdery droppings from infected birds. Now, if the birds are carrying the disease, that is the pathogen, and their droppings, if fallen on our body somewhere or something like that, they can generate the disease by the, again we'll have to inhale that from the dried droppings obviously, but this is how the transmission is possible. So on the list we have Legionnaire's disease, some allergic alveolitis, aspergillosis. These are definitely fungal in nature, but these are in the bacterial category. So we'll be discussing about some of these definitely in our later topics. Now we will be moving into the actual disease of whooping cough, the major, the most important, or rather causing a lot of death for the children or the infants. Now, whooping cough is characterized because of the typical whooping sound that occurs when the patient coughs, and it attacks mainly the children. Now, the organism is Bordetella pertussis, which you can see here actually are gram-negative coccobacillus. They are neither rod, neither cocci in typical sense. They are in between and they are gram-negative in nature. They are aerobic, that means they are respiring in the presence of oxygen. And they belong to the Alkaligenaceae family, and they are very, very specific to humans. The species Bordetella pertussis is actually infective to humans only. And their role is actually for colonizing the respiratory tract, and they bring about production of several toxins which bring the complicacy in the disease. Julie Baudet and Octave Gengu were the first ones who actually in 1906 found out a medium where these organisms could be cultured. Actually, it is difficult to culture, but they have the Baudet-Gengu medium devised, and the first serological testings were also carried out by them. So they have a good name in the history of Bordetella pertussis. To end the list, we have several others like Dr. Louis W. Sawyer, who in 1920s developed a vaccine for whooping cough. So this was the first time a vaccine was developed. In 1925, we have Thorvald Madsen, who actually tested a whole cell pertussis vaccine on wide scale. So this was the first human trial which was done, that is in 1925. And in 1942, the whole cell pertussis vaccine was actually combined with diphtheria and tetanus toxoids, and the first DTP combination vaccine was given in 1942 by Pearl Kendrick. Later we have Yuji Sato and others who have tried to produce other varieties of acellular pertussis vaccine. Okay, actually the whole cell means the whole organism is there, while acellular means some parts of the subunit vaccine usage is there. Let us see how exactly or what features in the organism brings about these kinds of disease. First, there are certain adhesions, meaning how organism adheres to the host cell. The three important ones are fimbriae, filamentous hemagglutinins, and the pertactin, which we can see here. This is the fimbriae and this is the filamentous hemagglutinins, and these are the pertactin. Basically, fimbriae and filamentous hemagglutinins are very important, and the fimbriae are meant solely for attachment, but it is not actually seen to be antigenic, or as such does not have much role in bringing about the disease as such. It is meant for attachment. While if you see the filamentous hemagglutinins, these have roles in serology, serological testing, and they are named because they appear filamentous in nature under the electron microscope. So we will be discussing mainly about the pertussis toxin in detail. Let us see the structure of it first. You can see here it is actually consisting of an hexamer or 6 units, while actually they are in the A and B form, meaning they have two centers, A and the B. Altogether you can see they are arranged in this fashion, and the receptor you can see here helps in the binding of the B subunit first to the epithelial cells of the respiratory tract. And then we have the A protomer, which is actually 27 kilodaltons. This causes the catalytic or enzymatic action, and that is what generates the changes that we will see, the coordinated action of several other toxins, which we will be briefly moving over here. The adenylate cyclase, this functions pertussinogen, is also responsible for the dysregulation of adenylate cyclase, as happens with the cholera toxin. I think you are aware of what happens with the cholera toxin. Yes. A similar effect happens, and we have the discharge of mucus and other things due to the dysregulation of adenylate cyclase AC, which functions to generate CAMP. Okay, so together with the pertussis toxin, the first one, PT, and AC, both help in the actual triggering of the organism into the ciliated cells. We will be seeing that shortly, but let us see what are the other toxins. The tracheal toxin is actually a peptidoglycan-like molecule. So basically the organism's cell wall is responsible for this, and it binds to the ciliary epithelial cells. The dermonecrotic toxin, on the other hand, actually causes the tracheal necrosis. The necrosis of the tracheal cells actually is brought about by the dermonecrotic toxin, introduced again by the organism Bordetella pertussis. The filamentous hemagglutinins, it actually binds, we have seen earlier, to the bacteria in the ciliated epithelial cells. And not to mention, lipopolysaccharide present in all gram-negative cells behaves as an endotoxin. So this has the fever-causing effect, as well as it causes inflammatory, activates the inflammatory cytokines and causes shock and cardiac arrest, like any other gram-negative cell can do. We can see here the coordinated function that I was just trying to mention. You can see the organism here with its tracheal toxins, filamentous hemagglutinins. So what actually happens in the ciliated epithelial cells, the normal function of cilia is to move, the beating of the cilia, which will, not let any organism move past down the tracheal cells and will throw up in the form of cough. But here, once the Bordetella has come, and because of several of its toxins and other fimbriae and attachment options, it attaches in between the ciliated cilia here. They generally rest here and entangles together, so gives rise to a ciliary stasis. Cilia do not move. And as such, these become stagnant, and we have the incorporation of more Bordetella into these cells and their growth and activity, causing the dysfunctioning of the actual working of the ciliated cells. So they will be growing and the toxin will be produced to bring about the effect. Next, we are moving to the symptoms. Okay, now symptoms are very, very important, and we can actually characterize the disease by the symptom itself. It begins with an incubation of 7 to 10 days where it is asymptomatic. There is no symptom during the incubation stage, where you can see the bacterial culture. That means if you plate, you will have the organism growing in numbers. You can see as the days passes. From the first to second week is the catarrhal stage. Here you have simple rhinorrhea, malaise, fever, some sneezing and anorexia. So some kind of discomfort is generally observed, and this stage is the maximum growing phase of the organism. Then is followed by the paroxysmal stage, where it is around the second till the fourth week, and this is the symptom which is shown by most patients, and then they are taken to the doctor. Earlier to this, it's generally like any other disease, but here you have the characteristic whooping cough, where the patient keeps on coughing and may at times have fits. Vomiting is also associated with it, and even at times you have the leukocytosis. Gradually then the organism count declines, and the patient moves to the convalescent stage, that is the recovery stage of 3 to 4 weeks, or it can be longer, during which there may be some secondary complications arising, pneumonia, seizure, encephalopathy, all these may be happening. So we have seen so far the symptoms. We have understood several agents which bring about the disease. We will now move on to the laboratory diagnosis. How can we diagnose the disease in lab? The first that is done is obviously culturing of the organism in a Bordet Gengu medium, which is a potato glycerol blood agar medium. Now this was devised by Bordet and Gengu, which has been already mentioned. Now the organism that should be present here should be of mercury drop pearl appearance colonies. These are suggestive of the Bordetella organism. What we can take is a nasopharyngeal swab. And we can also go for polymerase chain reaction in later cases, go for direct immunofluorescence study, or even serological methods later. Otherwise, if we are going for microscopy, simple microscopy, we'll look for gram-negative coccobacilli, can go for fluorescent antibody stain, and slide agglutination test can be done for serology.
Ma'am, I have a question. If we use antibiotics, what kind of antibiotics can be used for this type of disease?
We can use azithromycin or erythromycin. These can be generally used. But actually speaking, even if you diagnose in the laboratory, antibiotic treatment is of basically not much value. You have seen how the organism clings to the ciliated cells. So they will not be easily eliminated. Only thing is the duration of the disease can be reduced. And we generally move on for vaccination, that is prevention. Rather than treatment, vaccination is the choice. And several vaccines have been there in the during the processing, but finally now what we are using is the acellular vaccine. We have seen that whole cell vaccines were used initially. Now it is combined with diphtheria and tetanus for the children, because the majority risk is with the children. Person with a higher risk can also be vaccinated, but unlike the children, they are actually coming under the immunization schedule.
Ma'am, if a person has already suffered whooping cough and there is another person who has been vaccinated, then in which case the immunity will last longer?
you all know that natural immunity is longer. So basically for the people who are suffering from whooping cough will have more immunity, longer immunity. It can be as long as 20 years, it has been estimated, while a person who is vaccinated can have immunity little lesser, around 12 years or so has been estimated. So mainly children are vaccinated within a very short time. They are given booster doses as well, so that they can retain the immunity at least till 6 or even more years. But it is recommended by WHO that people who are working in some labs where pertussis can be a problem, they can also be vaccinated, and they should ask for combination vaccines during the tetanus vaccinations. So we have so far understood about the Bordetella organism. What is it? What does it contain? Which finally brings about the disease condition, seen the symptoms of the disease, some mode of prevention, antibiotics which can be given, which may not be of much help. And I hope you have understood or gained some importance as to how a respiratory tract infection can be caused by airborne transmission.